hurry and introduce our next researchers who specifically requested that they would follow Paul in their presentation because they have been studying this very issue in the mice model. And so I want to introduce to you Dr. Geoffrey Pitt and also Dr. Michael, I'm probably saying it wrong, Matsumi. They both come to us from Dr. Pitt's laboratory at Cornell. And just for the sake of time, I think I'll not give all the background information of their education and what have you, and we'll just go forward with their presentation. I know you will find it fascinating because they have mouse models of TS1 and TS2. For you atypical Timothy syndrome folks, we aren't quite there yet to specifically look at each of the changes in the calcium channel gene. I hope that's on the horizon, and people like Geoffrey Pitt and Michael will be those that will be able to give us some of the answers specifically on some of these variants. So Paul and Jeff and Mayako, you're on.
I'm gonna share my screen first.
Okay.
Can you see my screen?
Yes.
No. Yes.
Yeah.
Okay.
We can see you. I don't know about your screen.
Okay. Let me.
Yeah, we can't see your screen quite yet.
Okay. Sorry.
Okay.
Now, good.
Very good.
Okay. Thank you, Catherine, for a kind introduction. First of all, thank you for inviting me and giving me this opportunity to present my work, and thank you all for coming to join my talk today. I am a researcher in Geoffrey Pitt lab, Weill Cornell. Our lab studies functions of ion channel and diseases caused by channel disorder. And today I'd like to share my findings concerning study of hypoglycemia in Timothy syndrome mouse models that we generated in our lab. I think in this audience, I don't need to explain a lot about what is Timothy syndrome, so I'm going to keep this introduction part very short. But this Timothy syndrome, TS, is a multi-organ disorder caused by a mutation found in the voltage-gated calcium channels, CAB1.2. And some of the representative TS phenotypes that we study in our lab include craniofacial defects, such as round face, flat nose, dysmorphic jaw, and syndactyly, which is fused toes and fingers. And most known feature is the long QT interval, which generate abnormal heart rhythms. Neurological problems are also common. For example, about 70% of the Timothy syndrome patients have some degree of autism spectrum disorders. Mental retardation and seizures are also frequently found, as the channel plays an important role in neurological activities. The main difference between normal channel and TS channel is shown here, is increased calcium influx into the cells. And those increased calcium influx causes lots of problems in the body where the calcium channel is expressed. And another devastating feature in TS patients is hypoglycemia, that is a low blood glucose level that Dr. Thornton explained. And some of your kids may be experiencing this according to published journal, about 40%, 35 to 40% of TS patients exhibit hypoglycemic phenotype. So severe hypoglycemia is life-threatening, and sometimes just giving glucose cannot save the patient. So we really want to understand what is causing the hypoglycemia in TS. So normal CAB 1.2 plays an important role in the heart muscle to move properly so that the heart can pump the blood throughout our body. However, when the TS channel, TSCAB 1.2 is present in the heart, the mutant channel causes heart rhythm problems such as long QT. And we know that this channel is present in the pancreas, and the CAB 1.2 helps insulin secretion, which breaks down blood glucose after meal. And so based on the previous studies, investigators, including us, hypothesized that TS pancreas secretes too much insulin, and that accelerates hypoglycemia in TS patients. So before I show you my findings, I'd like to walk you through this diagram so that we all understand better how glucose is regulated in our body in response to decreased blood glucose levels. So I told you that insulin breaks down sugar after meal and reduce blood sugar to keep our blood sugar level in normal range. So the insulin regulation in the pancreas is important to maintain normal range of glucose levels. But what will happen when blood glucose levels falls very low? I want you to remember that there is another important glucose sensing organ, which is the brain. When blood glucose levels fall, the brain also senses it and sends molecules, signaling molecules, to the adrenal gland. And the adrenal medulla produces epinephrine, which is also called adrenaline. And that promotes glucose production in the liver through muscle, kidney, and fat. So all these pathways that are important for bringing back the foreign blood glucose to normal level are called glucose counter-regulatory response. So now we know how blood glucose is regulated, and the counter-regulatory response is important to maintain normal blood glucose level. However, several studies of diabetic patients and mouse models show repeated hypoglycemic episode cause impaired counter-regulatory response. When diabetic people or mice experienced multiple hypoglycemic episodes, adrenaline release from adrenal medulla was reduced, and adrenaline is critical to promote glucose production in the liver and from the other organs such as muscle and fat. But when the glucose counter-regulatory response does not work properly, now normal range of blood glucose cannot be maintained. So our research seeks to understand how hypoglycemia in TS patients occurs using TS mouse model. To study Timothy syndrome, we made both type 1 and type 2 Timothy syndrome mouse models using the latest gene editing technique called CRISPR-Cas9, which received Nobel Prize in Chemistry this year. These mice have the same mutation as TS people have. Type 1 Timothy syndrome mouse model, which we call TS1, has a mutation in exon 8A of CAB 1.2. On the other hand, type 2 Timothy syndrome mouse carries the same mutation in exon 8 of the same gene. So these mice carry TS channel that allows too much calcium influx into the cells as TSP will do. Okay. So using these mice, we first checked if TS mice have abnormal baseline glucose levels. First, I checked TS1 mice, and they showed normal blood glucose level. So wild type is normal mouse, and TS1 also has the same level of blood glucose levels. On the other hand, TS2 mice showed lower baseline glucose level. The normal blood glucose levels in TS1 and hypoglycemic phenotype in TS2 mouse models may be because of the differential expression of splice variant exon 8 and exon 8 between mice and humans. And another indication of differential expression of the splice variant exon 8A and exon 8 between mice and human is the syndactally fused toes. When we looked at TS1 mice, we didn't see fused toe here. And TS2 also didn't have syndactly. So since we had the TS2 mouse model, which has a hypoglycemic phenotype, we decided to study TS2 mouse model for our hypoglycemic project. So here I'm showing you is a glucose tolerance test, GTT. GTT is to see how fast or slow the body can clear blood glucose. This test is sometimes given to pregnant women to test pregnancy diabetes. And in this test, mice are fasted for 16 hours, and then they were injected with glucose to see how blood glucose levels changes over time. So after giving glucose, we measured glucose levels every 15 minutes. And this y-axis shows blood glucose level, and this is time. So if you look at blue line, this is the wild type, normal mouse. When they are given glucose injection, the blood glucose levels going up and then slowly going down and go back to normal blood glucose levels. But when we gave glucose to TS2, they cannot raise blood glucose level, and the blood glucose level in TS2 stays very low all the time. So these results indicate that the wild type mice can raise blood glucose level and maintain normal level over time. However, blood glucose levels in TS2 group stays low. So next, we did insulin tolerance test, ITT. This is to see if counter-regulatory mechanism is working in the TS2 mice by generating artificial hypoglycemic status. Here, the mice were fasted for 3 hours and then injected with insulin to see if blood glucose level can recover using the counter-regulatory mechanism. And this time, we also measured blood glucose every 15 minutes. So after fasting, the normal mice showed a little bit higher glucose level. And then when they were injected with insulin, the blood glucose goes down, but counter-regulatory response kicks in, so they can recover blood glucose levels over time. However, TS2 mice showed lower baseline blood glucose level after fasting. And when we gave insulin to them, the blood glucose went down, and then they couldn't recover the blood glucose level. So these results suggest mutant counter-regulatory response may be blunted. We also wanted to know if mutant liver is working properly so that they can produce glucose in the liver. To test this, we performed pyruvate tolerance test, PTT. And in this test, mice were fasted like ITT. And then this time we gave pyruvate. And again, we measured glucose level every 15 minutes. And as you can see, both normal mice and TS2 mice can raise blood glucose after receiving pyruvate. So these, in this test, we see both wild type and mutant mice are raising the blood glucose. This means that TS2 mouse liver can produce glucose. So till now, in GTT, I showed you glucose clearance is abnormal in TS2. And in ITT, after insulin injection, TS2 mice cannot raise blood glucose level, suggesting that blunted counter-regulatory response. And in PTT, we saw normal blood glucose, indicating TS mice have normal glucose production in liver. So what is causing these problems? As I showed you earlier, based on the previous studies in which people found that CAB1.2 is present in the pancreatic beta cells and promote insulin secretion, other people, and we hypothesized that TS channel in the pancreatic beta cells secrete too much insulin, and that is causing hypoglycemia in TS people. So to test this hypothesis, our collaborator Nico from James Low Lab at Weill Cornell performed glucose-stimulated insulin secretion assay. In this assay, he isolated pancreatic beta cells from the wild type or TS2 mice and directly exposed them to glucose to see if insulin secretion is elevated in TS cells. And he tested different concentrations of glucose. But contrary to our hypothesis, this assay showed no glucose elevation in the beta cells from TS2 pancreas in response to glucose stimulation. So this graph shows this is normal cells and this is TS cell. And we expected this to be higher than normal beta cells, but we didn't see that results. And this was really unexpected for us. So we had to confirm these results in other ways. And to do so, we generated another mouse line. This time, TS channel was expressed only in the pancreatic beta cells. We chose this method so that we can see the mutant channel effect only in the pancreatic beta cells. So we made the mice and performed the glucose tolerance test, GTT, using these mice. And measured glucose level like before. And as you can see, between normal mice and the mice with calcium channel only, the mutant calcium channel only in the beta cell of pancreas, both of them showed normal blood glucose level after GTT. So this is consistent to Nico's data, and all these data suggest that the major cause of hypoglycemia in TS may not be because of hyperinsulinemia, which is elevated insulin caused by TS channel in the beta cells. So this was surprising, but the question remained, and what then is causing hypoglycemia in TS if not the pancreas? As I showed you earlier, there is another important glucose sensing organ, which is the brain. The brain can also control blood glucose level. And it is known that the region called hypothalamus in the brain, indicated in yellow circle, plays important roles in various metabolic regulation. And we confirmed that CAV1.2 is more intensely expressed in the specific regions of hypothalamus, as shown in green dots. These neurons are also known to control glucose homeostasis. So this time, we activated TS calcium channel only in the neurons located in hypothalamus, and again performed GTT. And we saw reduced glucose levels, shown in red line, in the mutant mice compared to wild type, which is blue line. We are currently repeating the experiment to confirm these results. And so these results suggest TS channel expressed in the specific region of the hypothalamus may cause hypoglycemia. So to conclude the hypoglycemia part of my talk, we found TS channel in the pancreas does not show elevated insulin, suggesting that TS channel in the pancreas may not be the main cause of hypoglycemia. We also found expressing TS channel in the brain causes lower blood glucose, indicating TS channel in the brain may be responsible for hypoglycemia. And it's not surprising that CAV1.2 in the brain causes all sorts of problems occurring in our body, as the brain is like a conductor of orchestra, sending a message to each part of the body to do proper jobs. And CAV1.2 is strongly expressed in the brain. In fact, TS mice display neurological disorders, including autism spectrum disorder like behavioral abnormalities in our TS mice. We see abnormal social interaction, repetitive behavior, and impaired social communication. In addition, they also show abnormal bladder function, abnormal heart rhythms, including long QT. And we also recently noticed that they seem to have exercise or stress-induced abnormal body movement under the stress or exercise. After exercise, they lose control of the body movement. So these mice are very precious and help for us to understand underlying mechanisms of TS-related features. And recently, through the communication with Catherine, we were told some of the TS patients suffer from constipation. So we wanted to see if our TS mice have this phenotype so that we can study the CAV1.2 is so that we can study that. And CAV1.2 is expressed in the smooth muscle of digestive tract, and Arvin in our lab performed gut motility assay. In this assay, mice were fasted overnight and given red dye after fasting. And then we measured how long it takes for the mice to poop red dye. And we saw that difference between wild type and TS. It seems like TS mice takes more time to poop. So hopefully this this result indicate that abnormal gut movement. So hopefully we will be able to report new findings in this area of study as well as hypoglycemia project at the next meeting. That's all. And thank you for your attention. With that, if there is any question or comment.
That was extremely interesting. Thank you so much. I know a lot of people may have some questions as we talked about constipation and all of course brain issues. So thank you so much. Does anyone have any quick questions before we move on to our next presenter? And again, please put any questions that you may have in chat and we can get those answered for you later on as well.
Can I just have a quick question? Or just a comment, because we just got some interesting findings from the, I don't know, biopsy or the tissue results of my daughter's intestines, because she had to remove a part of her intestines and have an ostomy placed.
Yes.
And in the bowel tissue results, they found like fibrosis and different like neural damage in the ganglions and like inflammation in the neuron cells in almost all the layers in her intestine. It's really hard for me to find the medical words when I'm not English, but I hope you understand.
Yes.
And if you want them, I can send them to you.
Yes, that'd be great.
Because they had to send her colon to England to get those results.
Oh, yes, it's interesting because I told you that the CAD1.2 is expressed in the smooth muscle, but it also expressed in the nerves, which connect brain to gut. So we actually don't know what is causing the problem. So it's very interesting. Jeff, do you have any comments on this?
I don't have anything else to say right now, but it is a very interesting finding. And as you said, Micah, we're still trying to dissect all the different places where this channel is expressed and how it might contribute. And I think that one of the things that we've learned recently by focusing on this hypoglycemia project is that the broad expression of the calcium channel is leading us to understand much more about the physiology.
I want to thank both of you for all of your excellent work. I've known Dr. Pitt now for several years. We've worked on a few different projects together, and it's been wonderful to meet Maiko and know that she is so expert in all of this research. I also wanted to mention to the group of your new paper that was just put out a couple of weeks ago. It was a review of voltage graded calcium channels in the function in non-excitable cells, which also highlighted our Timothy kids for bone development. And gosh, Jeff, we so appreciate all that you're doing. My goodness, that's just wonderful. I'll send this to our website or our Facebook page so that if people would like to read some of that, they can. But please, please know of our appreciation for all that you're doing, your good works. And I'm sure that this particular slide presentation will be of help to Paul and some of his thinking about Timothy syndrome. So I'm so glad you recommended that you come after, so that it could be in explanation, possibly to help in some of his thoughts. Okay, well,
Thank you.
thank you both very much, Mako. It's wonderful to see your face again.
Thanks.
Take care.
You
Take
too.
care of your new baby.
Thanks.
All righty, we'll go on